Thank you. So it's my pleasure to welcome Bharath Ramasundar, who is the founder and CEO of Deep Forest Sciences. He received his PhD in computer science from Stanford University as a Hertz Fellow, where he studied the applications of deep learning problems to drug discovery. He created DeepChem.io, an open source project to grow the deep drug discovery open source community, co-created the MoleculeNet.ai benchmark suite to facilitate development of molecular algorithms. He's one of the primary developers of Camberta, one of the foundation models for small molecule property prediction, and we'll let him take away. Thanks, Bharath.
And thank you very much for the intro. I will also just quickly, apologize to everyone in advance. I have a hard stop at 4 p.m. Eastern. We should be able to get through the slides and still have some time for questions. I might skip through a couple of things and point them at references, but I should be able to share the slides after. Okay, first, I think I can skip through this. Just a couple of things I worked on. I think Arvind already covered that. As Arvind mentioned, one of the main things that I did in my PhD and in the years since has been developing an open source framework called DeepChem. DeepChem tries to make it easy to apply scientific machine learning to various problems. One of our first applications really is, I'd say, one of the first high quality open source implementations of molecular graph convolutions. Since then, we've added a broad variety of different models. For example, Camberta was developed under DeepChem, and as well as several new models that we've been adding in recent releases. It's a broadly used package. Lots of folks use it. All the institutions here have written, say, either a paper or blog posters like using DeepChem. And it's a large and actively growing community. So at the end, I'll have some information about how to, say, join our community Discord and things like that. But if you're interested, please do join in the conversations there. All right. So with those, preliminaries out of the way, I thought it might be useful to just set the stage a little bit and talk about, one particular application area in drug discovery. So I know for most of you are working in different scientific fields, so hopefully what I say won't be too much of a repeat. I think Philippe covered some of these themes already in his talk. Really great stuff that, a lot of the things I think I'll say will broadly echo things that Philippe is saying. And I'm hoping at the end I can also maybe share just a little bit of the experience that we've had from applying some of these techniques in practice and industry on real world problems and how that can feed back into research applications as well. So for context in drug discovery, there's one of the hardest challenges that you face is that you are trying to design molecules that do new and interesting things, but it's a very fuzzy type of development in that many times you don't even know precisely what property you're optimizing. So you have to first start with, say, some hypothesis about the disease. That's the first step. Once you have that hypothesis, you have to find some molecule that, say, can modulate, say, a protein that you posit is the therapeutic target. From there, you have to systematically optimize the molecule you've created. In this talk, we'll focus a lot on what are called small molecules, but there's a broad range of other modalities people have come up with in recent years for how to modulate, say, potential biological systems. And then at the very end, you get one compound out. Now, part of the difficulty here is that there's no real mathematical or even experimental model for many of these diseases. The only real source of ground truth is a clinical trial, and a clinical trial is extremely expensive and extremely slow. So in many ways, I think it's a very challenging scientific problem because the number of true data points that you have is extremely limited. This has been borne out one thing that a lot of people in the industry like to talk about is something called Eroom's law. It's the opposite of Moore's law, spelled backwards. It's just a observation that the cost to find new medicine has been steadily rising over the last several decades. I think the last few years there's been reforms, so it's kind of plateaued at the bottom, but it's not really getting cheaper to find a new drug despite many years of new methods, including AI. And I can, at the end, maybe share a couple of my thoughts about why I think that's the case and, maybe ways we can do better in the future. But coming back to the science, sorry, I apologize. I will skip ahead a little bit just to a slide because I accidentally put one out of order. Coming back, a little bit here to the science of drug discovery, part of the fundamental challenge is you're dealing with a very low data problem. At the end, the only real sources of ground truth are what you get out of clinical trials or actual human data. that's a very, very limited pool. And almost by necessity, the data you get is from drugs that already are known to work. So we're designing a potential drug for a new disease area or new application. You're basically dealing with a system where you don't have that much scientific data. So in given very low data, how can you actually do meaningful science? And again, this is a big field. Lots of people have been working on it. So I'll try to give you a couple of things that I've seen personally and I've learned, but I think it's also useful to think about this because I know many of you are not working drug discovery as a case study for how you can do machine learning in a context of very high scientific uncertainty. And I know in other applications where you're trying to do new scientific discovery, there's variance in this, right? We don't have room temperature superconductors today, so you have to say rely on theory to get you there. So, jumping back a little bit here, just to some basics of machine learning in the field. Like the first step in how you do machine learning on drug-like molecules is you first turn them into vectors. This is, old school stuff. Like people have been doing this, I think, since the 60s and 70s when they first started putting molecules in databases. In more recent years, I'd say in the last 10 years, the shift has been towards, using first graph convolutions, where the idea there is, again, um, if you can adapt some of the thinking that has been working in convolutional networks over to molecular structures by treating them as mathematical graphs, you could potentially get some interesting properties. So what I'll say about graph convolutions is that um, they are a really cool technique. They tend to work well when you have enough data. Um, when you don't have enough data, though, the old school techniques of just creating these hand-coded fingerprints actually can be very robust. Like the graph convolutions only really, I think, have a market advantage when there's sufficient data. So they're a powerful tool, especially in cases where we have lots of experimental data, but not really a fundamental solution to this low data problem in drug discovery. And now, in the last few years, I think that, as Philippe mentioned in the previous talk, there's been, large growth in the use of transformers. So I think, Philippe, of course, and his group did some of the early work here in the space. And I think, we picked up from some of Philippe's work as well with our converter models. And the idea there is, again, you treat the smile string as basically a sentence, you tokenize it, you feed it into a transformer, and then you do things with the downstream. And from here, I think that there's like a couple of different avenues you can take. One way is, of course, you can try to go more broad sweep to just using the large LLMs that have really taken, the AI community and the scientific community by storm. But I think, another approach, which is more the one that we've followed, is saying, can you change the way you think about pre-training in order to have richer physical priors baked in? And I think there's a couple reasons for this. The first, of course, is that training a big LLM is hard. And if you're committing to using a big LLM, you also then need to do a lot of hard work to make sure you have the inference capacity to even be able to fine tune it. We're a small team. It's hard for us to work with large LLMs at times. We've done experiments with, say, various, I think GPT-4, Llama 2, etc. And while the LLMs are getting better, say with the latest releases like 4.0, Claude or things like that, it is still quite gnarly to use these things. Whereas a smaller model, even a smaller transformer, can be much easier to work with. So there's a notion of just convenience and training and how you choose to work with these. So again, like this will be a little bit of a slightly different talk from, say, the theme of like, using the big LLMs that Philippe was touched on, but I think hopefully it'll be a useful complement to some of the things that you all have already learned about from other speakers. So, jumping ahead, part of like going back to the fundamental problem of low data that I just mentioned, there's a wide variety of strategies you can use to tackle this low data problem. Like a lot of them I won't cover in this talk, but other things I'll just briefly mention. You can augment your training data with physical simulations. You can use ideas from meta learning and metric learning to make the most of low data. You can do multitask learning, or you can use chemical foundation models. And here, the basic idea, as with many other applications of self-supervised learning, is that although supervised labeled data is expensive, you can find a lot of unlabeled data if you get creative. So if you look at, say, available, quote unquote, labeled data, these are coming from experiments. And the one note I'll make here is that it's important to remember that even these experiments are proxies for, the underlying clinical trial. So the experiments themselves, you can only think about as like very noisy labels to an underlying scientific reality. But that said, they're a lot better than not having experimental data. The challenge, though, is that even for the largest scale experiments, you tend to have limited data when compared to, say, web scale data that you see in other contexts, or even in other scientific applications. There's just not that many small molecule measurements out there. And part of the reason is that unlike, say, other structured classes of biomolecules like proteins or DNA or RNA, small molecules are more, unstructured and freeform. So there's not like a simple combinatorial set of reactions that can let you build all small molecules. There do exist families of friendly reactions that do easily, form, what's called quick chemistry, are able to actually combine them, recombine them. But a lot of the time, you have to break out of these spaces of molecules in order to test different hypotheses. So it's fundamentally a problem where it's hard to generate data until someone, say, invents something like the universal synthesis machine. And I think that's going to be true for a while. There's a lot of groups working on that if you're curious, but I won't cover that in this talk. But what you can do, going to this click chemistry that I just mentioned, is that you can use like large vendor catalogs of unlabeled data, where different scientific vendors have said, we've studied this family of reactions well, we think we can compose and recompose them. And here's a collection of, say, 5 billion or 10 billion, or in some cases, 50 billion molecules that we think that we can make in-house with high confidence that we believe follow from these combinatorial reactions that we've already covered. So coming back to the application of self-supervised learning, although this unlabeled data is purely, say, in many cases hypothetical, these molecules don't necessarily exist. They're high enough fidelity that there is some information we can pull out of them. And if we can use this information to bootstrap the available labeled data, perhaps we can do better and have a systematic approach to dealing with low data in drug discovery. So how do you do that then is the next question. So here's a slide, borrowed from one of Jan LeCun's talks. I really recommend the talk. I have the link here for anyone who's interested. Like you come to a question of, what is self-supervised learning? And I think, Jan LeCun has a wonderful definition here. It's filling in the blanks. You're taking a signal, you're occluding part of the signal, and you're asking the model to learn to fill in the blanks. This could be predicting the future, can be predicting the past, can be predicting parts of the present, or some combination of all of the above. And when you take this principle and apply it to molecules, you say, well, how can I actually do this type of self-supervised learning for molecular structures? And the answer is this is a very active area of research. There's a lot of people exploring different methods for doing self-supervision on molecules. I'll briefly cover a few different approaches that we and other groups have tried, but I'll say it's also, it's very much the case that I think it's an unsolved question how you do self-supervision well on molecules. And at the end, I'll also come back a little bit to bigger, broader LLMs and talk a little bit about how some of the self-supervision strategies that we've covered today compare with, say, strategies that people are using for the big LLMs. In brief, though, there's a few different approaches that I think are common in the literature. One is, say, property-based predictions. So you say, given the structure, predict various chemical properties. These can be things like, molecular weight, number of rotatable bonds, or it can be like infill properties, like if you remove an atom from a molecule, can you predict the missing atom? Other approaches, well, actually, it's the second one. I covered the infill approach. The other approach is, graph-based approaches where you're looking at things like mutual information. So there's a lot of creative, oh, sorry, is someone saying something? All right.
No, go ahead, Bharath.
There's a lot of creative work people have been looking at with different mutual information-based scores for maximizing the information that can be extracted from different parts of a molecular structure. Again, I think this approach is to maybe jump ahead a little bit. Really interesting stuff, a little mathematically, very mathematically novel, but maybe not yet as mature as some of the other approaches I'll cover. And I think that there is hope for, say, combining these different approaches to get something out that does actually capture more the reality of how molecules actually operate. But even then, I think, again, this is a very active area of research. I think there's a lot of questions here that still remain to be answered. All right. So starting with, approach one, the basic idea is you take a molecule, you then convert it into a smile string. And then from the smile string, what you do is you can predict basic chemical properties. Again, these are things you can compute using standard chemoinformatic toolkits like, atomic weight, number of carbons, number of polar bonds, et cetera, et cetera. Or you can pluck out certain missing, atoms or bonds from the molecule and then train the model to do an infill. So the strategy of filling in the missing atoms and bonds, it's often implemented by using, say, a standard NLP-based pipeline. So you just pretend that the input small molecule is just a sentence, and then you do, masked language modeling or tokenization on the tokenization. So there, the nice thing is you can just plug into existing, say, Hugging Face pipeline that you're using for other contexts. So that's very nice. Whereas for the predicting basic chemical properties, you do need to build a more custom pipeline. DeepChem has tooling to make a lot of this easy for you. But if you are looking at it, it won't be, say, like your standard Hugging Face pipeline. You probably need to use something like DeepChem or roll your own package or framework or pipeline for doing that. Now, going to say some of the other approaches that I mentioned briefly, like there's a lot of in the chemoinformatic literature, there's been a long-standing debate over, two-dimensional versus three-dimensional information. Part of the challenge here is that, molecules are fundamentally, dynamic three-dimensional entities. They change shape. The shape is often very important for how they're how they actually behave in practice. And if you treat the molecule purely as like a two-dimensional graph, are you losing information? And there's been a lot of back and forth, and I don't think even now there's like a clear understanding of like when 2D is better, 3D is better. We have some decent heuristics that work in practice. But I think one of the neatest ideas I've seen the last couple of years has been a notion that you can use mutual information as a way to train a two-dimensional representation to recapture some of the information that's available in three-dimensional representations. So that, again, there's a really neat idea of like information retrieval or regain, where even though you stay with the 2D information, you can train the model to effectively learn to predict the 3D structure. So it's almost like you have 3D structure prediction as a little submodule implicitly in these networks. Again, I think it's a really neat idea mathematically, which is why I often put it in these slides. But it's something I wouldn't say is quite ready for prime time usage. We've done some internal benchmarks trying to use this approach. It works decently, but it's not, say, quite as robust as, say, the chemical property prediction methods that we covered previously. There's other mutual information-based approaches. Like this one, I think is interesting. It tries to maximize the mutual information between local and global parts of a graph. This figure, borrowed from the original paper, is a little bit opaque, but the idea, as explained, is if you treat a molecule like a graph, you have like local atoms, which are nodes in the graph, and you also have the global molecule. And if you can make it so that the global representation is incentivized to maintain the local information at each of the atomic units, that gives you a better picture of the global behavior of the molecule. And you do this by maximizing mutual information score. Again, this is a nice, elegant idea. It's very cheap to implement. There's implementation DeepChem if you're curious to try it out. But again, I think these ideas, these graph-based self-supervision as a field is, I'd say, still less well developed than, say, the transformer-based self-supervision. So hasn't found as much uptake yet in the literature, but I think it's one of these ideas that it's a matter of just like there's something neat here, but it just kind of needs to mature to a point where it can be useful in practice. All right, so now maybe returning to transformer-based representations. As kind of mentioned earlier, one of the papers that got us really excited about using these transformer-based representations is a project we did a few years ago called Camberta. In Camberta, we took, again, simple transformer on Smiles strings. We plugged in, say, a database of these Smiles-based compounds, and we did a simple scaling experiment. And part of what got me excited was just that seeing how the scaling improved downstream performance. It was really exciting because the type of improvements that we were seeing matched in magnitude some of the improvements that we'd seen from these other approaches that I mentioned earlier, like multitask learning or mutual information-based approaches or the like, but in a very simple manner that let us reuse, say, standard open source pipelines. Now, of course, it turned out in future work that the story is a little more complicated than that, but as a proof of concept, it was something that got us really excited about exploring the potential for these chemical foundation models. In subsequent work, I think we did like in follow-up, we scaled this up a little bit larger. So the first converter, we only trained up to 1 million molecules or so. In the second iteration of the series, in Converter 2, we took this up to something like 80 million molecules. It's a couple of years ago now. And we came up with a strategy in particular for sharing hyperparameters tuned on smaller proxies when actually tuning models for the larger full data set. So what we did was we actually just reused hyperparameters between different learning objectives. Like the mass language modeling I mentioned earlier is fairly cheap as an objective as opposed to the property prediction-based predict chemical properties approach. So what we found doing some basic testing of this work was that sharing hyperparameters didn't seem to damage the performance that much. So that turned out to be a really useful strategy for scaling this out. At the time when we put this work out, again, a few years out of date, I'd say Converter 2 was about par with, say, DMPNNs from Chemprop, which is, again, some of the state-of-the-art methods that were out there. So again, it was another proof of concept that applying these methods in practice was possible and could achieve, state-of-art performance matching, say, the best graph convolutional or random forest model that was out there. So again, for us, it was an exciting proof of concept showing that you can use these transformer-based approaches in practical chemoinformatic problems. Now, from there, before I move on, I'll mention we've been working on Converter 3 for a few years now. I think we should have something out probably on archive later this year alongside an open source release to just give a couple of ideas about why it's taken us a couple of years to actually get the next iteration of the series out. It's quite hard as a small team to train a big LLM. We had to build a lot of infrastructure to actually be able to train models ourselves. So our latest approach, we have, I think, models training up to 128 GPUs, but it took a lot of infrastructure building on top of DeepChem, and we'll put out a repo with the paper release that showcases some of the infrastructure that we had to build to make it happen. But what I mentioned is that for some of you, especially if you are from a smaller academic group or a smaller company, and you're trying to build your own foundation models, it's really hard. There's a lot of classes of failures that we ran into, like different node communication failures, like random restarts, we'll have to go back and actually write down some of the class of failures. And again, the big tech companies have seen and cleared a number of these failures already. You can say, I think Facebook has some excellent reports on some of the class of errors that they saw when training the Llama 2s. But again, it's, I think, still useful in that a lot of these errors still come about when you're training even relatively smaller models for chemical applications. But again, I think that my recommendation would be that if you're an academic, if you can get a grant that gives you access to supercomputing facilities, or if you can partner with another institution that can provide that, it's worth it. We took a hard approach of trying to build up our own open source infrastructure. We think it'll be quite useful to the community when we put this out, but it was a much longer, harder path than I would have guessed when we first set out at doing this. Again, I won't say too much yet because the paper isn't out. But there's a lot of infrastructure, last 2 years of DeepChem development that has gone into enabling Converter 3, and that should be out later this year. And finally, just wrapping up the talk, I'll pivot a little bit and just talk a little bit about some of the strategies that we found have been necessary to apply some of these methods to real-world drug discovery. Unfortunately, a lot of the work that we do is, very strictly under NDA, so we can't talk about it. So instead, I'll talk about carefully selected proxies that I think embody some of the same ideas. Active learning. Here's a paper from a couple of years ago on a toy problem. We showed that active learning would speed up how quickly you can get interesting drug-like molecules. A lot of these strategies actually work well in real-world practice as well. So we've applied similar strategies on actual drug discovery problems. Non-toy, I cannot share those results with you, but I think being able to have an active learning strategy, and what we find works in practice, is keep it simple. We don't try to use fancy Bayesian things or things like that. It's often very dead stupid, but dead stupid often is very robust to real world practice. So that's worked well. Again, as a collaboration.
So there, the active learning loop in the previous, so this is you're running MD simulations, or how are you generating the data?
This particular paper, this is purely a toy data set. So there's again, it's a very simple toy. It's just a proxy model.
Okay.
In the real world application, it's there's an experimental loop where actually it's going out to an assay and generating data.
Okay.
And We have done some with molecular dynamics, but the industrial use cases are all like, they need an experimental readout, and it's like different assays they're using for that.
Okay, thank you.
And again, this is a collaboration, Venkat. Again, I think that so like showing this as a public proxy of the type of like, I think having these types of design loops where you can slot in a machine learning model into one part of a larger pipeline, that's been really important for us. A lot of the pipelines we build, there's multi-stage like cleanup, different simulations. The machine learning is important regardless of what model you use, but it's like one part in a broader picture. And again, I think this diagram thematically is very correct in that this is like how we found machine learning to actually slot into projects in practice. It's an important subcomponent, but it is just like 10% of the whole when you're trying to build even a computational pipeline for a large project. So again, thematically right, but we can't share many details about the practice. And since I apologize again, I have a hard stop in 5 minutes. I'll just quickly mention a couple of things that we've seen from different use cases. Again, we've done various internal experiments with large language models. What we've seen so far, and I think Philippe touched very nicely on a number of these themes, is that they can be really fragile. Like it's really hard to tell what they understand and don't understand. When we tried to do fair, quote unquote, fair comparisons using, say, like some of the same scaffold-based splitting that we would do on chemoinformatic models, the models did really poorly. And there's an argument that we're being too hard on the models, and these we're testing on things that they haven't been trained on. But the challenge then you have is like for a lot of these models, you have to say use GPT-4 or Claude or whatever, and they've done the training. You don't really know what's in there. So it's really hard to do science on these models because you don't often have control of what's in there. And that's part of the reason why we set on a path of trying to build our own training infrastructure. But again, much harder, more painful path than I would necessarily recommend to anyone else right now. So I think there's still some hard problems before you can try using LLMs on practical drug discovery problems. People get really excited because it feels like you're talking to the machine. And it's often, but it's really hard to control for like, when is the human leaking in information to actually get good results out of it? But people get really excited. I think there's something there, but I think it's going to be hard again to see real results come out. And it's going to take a lot of hard scientific work. Hopefully that some of you in the room will lead. And finally, again, I already covered a lot of these themes. I'll skip over in the interest of time, but I thought it might be useful just to distill a couple of lessons that we've seen in practice. Again, chemical composition models can be really useful, but I'd say, takeaways like in practice, not trusting machine learning too much is often a good rule of thumb in scientific discovery. Sometimes you see very popular machine learning models very overfit, and when you test them on systems that are outside what they've seen, they tend to break really badly. Simple physics-based approaches can work pretty well in new scientific discovery contexts. With that, I'll end there because I have to hop out in a couple of minutes. I at least want to be able to take questions if there's any.
Thank you very much.
Questions?
Bharath, in that scaling plot that you showed, right? At what point at what data set size do you think these models will start to beat the purely graph-based models?
So I think, there's been like IBM's Molformer, there's been a number of models, NVIDIA's MegaMolPart, that they claim are beating, say, the graph-based approaches already. We've not been able to fully replicate these results, but that's probably a limitation of our training infrastructure. So I can believe that they already are beating the graph-based methods at sufficient scale. But again, we want a good, strong open source reproduction that we can trust. We haven't gotten there. That's the only part that I think that there's work to be done. That's what we're trying to do with Converter 3.
Questions?
Yes. Oh, sorry. So I think yes, we can share. I'm happy to share these slides over. I'll coordinate, I think, with Venkat, and I do apologize that I have to hop out in a couple of minutes. Is there anything that?
Yeah. Thanks, Bharath. Yeah.
Thank you very much for your talk.
Pleasure. And again, like maybe the last thing I'll say is, again, I think for a lot of these like real world applications, a lot of what we've seen is it's very tricky to get machine learning to actually do useful things. 95% of our work is not machine learning related, despite being a machine learning company. And again, this is probably true for many of you who've done this in other pipelines, but I just want to share that I think machine learning and scientific discovery is still a ways to go. But thank you for the chance to present to you all. I'll send these slides over. And again, if you're interested, please join our community Discord. We have a lot of folks talking about machine learning and chemistry on the DeepChem Discord. All right, thank you, folks.
Thanks, Bharath.